Well, the date and the time is here. Hello, and welcome to today's session on Project Optimus. I'm Arlene Padron, Director of Marketing for Simulations Plus, and while we have over 1,300 registrants logging in, I want to ensure we have plenty of time for questions, so I'm quickly going to run through a few housekeeping items. We take your privacy rights seriously, and by attending this event, or participating in the Q&A session, you're allowing us to contact you for follow-up. You may ask questions today throughout the entire presentation, and Tom Sun, today's moderator, will review those and have them ready for our speakers today. Use the Q&A tab on your control panel, and if you have any questions, feel free to drop them into chat if you need technical assistance. A recorded version of this webinar will be available for replay and for you to share with your colleagues. So for those of you just joining in, welcome. While our presenters and audience are settling in, I'm going to post a quick poll for you. What's your primary interest in today's webinar? Go ahead and pop your answers into the chat poll. I'll give you just a couple of seconds here. All right, I'm going to end the poll now. And it looks like your goal is scientific education, which I am sure you will get plenty of today. So without further ado, I'm going to turn it over to Tom Sun, today's moderator and Vice President of Strategic Consulting Services. Dr. Sun has over 20 years of experience in the pharmaceutical industry, specializing in the application of model-informed drug development in oncology. Tom, take it away.
Thank you, Arlene, for your introduction. Again, my name is Tom Sun. I will be your moderator for this section. I'd like to welcome you to join our September 2022 Simulation Plus webinar. Today's topic is Project Optimus, FDA's new dose optimization and selection paradigm in oncology drug development. It is our pleasure to invite two distinguished speakers from FDA. Our first speaker is Dr. Brian Booth. He's the Director of Division of Cancer Pharmacology 1, the Office of Clinical Pharmacology from the FDA. Dr. Booth received his PhD in pharmacology and toxicology at Queen's University in Canada, postdoc training in pharmaceutical sciences at University of Buffalo, and joined the FDA in 1998. He is involved with the clinical pharmacology development of several hundred new drug applications for oncology drugs. And along his career has been teaching new clinical pharmacology reviewers and medical officers. Dr. Booth has a distinguished career and excellence in publishing more than 80 peer-reviewed articles and book chapters. We also want to welcome our second speaker, Dr. Hao Zhu. He is the acting director at the Division of Pharmacometrics, the Office of Clinical Pharmacology at FDA. Dr. Zhu received his PhD in pharmaceutical sciences and master degrees in statistics at the University of Florida. He started his career in modeling simulation in Johnson & Johnson and Bristol Myers Squibb, and he joined FDA more than 15 years ago, and he has served as clinical pharmacology team leader and a QT IRT scientific lead. His division at FDA reviews the pharmacometrics-related submissions and support pharmacometric-related policies in the agency. And as a quick reminder that we encourage you to submit your question to our speakers throughout the presentation through the Q&A tab on your screen. And we'll discuss the selected questions at the end of presentation and the final poll questions. So without further ado, here's our first speaker, Dr. Brian Booth. Thank you.
Well, thank you, Tom. And on behalf of Hao and myself, I'd like to thank you for the opportunity to be here today to speak to everybody in the audience about this topic. And we're looking forward to this as an engagement to get to better, more successful strategies to selecting dose. So what we're going to do, let's go here. There we go. So what we're going to talk about today, I'm going to spend the first part of this presentation to talk to you about Project Optimus, give you an overview of this project and why we're so interested in this now. And then I'll get into a case study with idelalisib to look at some of the lessons we might learn about selecting doses better, which will lead us to the concept of dose optimization. And then I will hand this off to Hao, and he's going to spend the rest of the time talking about the MIDD tools that are available to help select doses, and he'll underscore those with a couple of successful cases where these kind of approaches have been used to get to those right doses. So Project Optimus is an Oncology Center of Excellence initiative that was started, I guess, about a year ago or so, formally. And the point of this is to find the doses for cancer drugs that are optimized. That is to say, we've maximized the efficacy and we have the lowest amount of toxicity that we can get to. Okay, and one of the important tenets of this is the one I've highlighted down here in yellow, at least it is to me. And that underscores the notion that we want to use all of the non-clinical and clinical information that we have early in development to really find the best doses early in development, rather than trying to deal with that later in drug development. So the question we're asking here, it's actually not a new question. We have looked at this for a long time. It is, in fact, the raison d'etre for the Office of Clinical Pharmacology. Okay? According to the vision and mission that is articulated by our office director, Dr. Zine, we want to apply drug response knowledge into the regulatory decisions that we make about doses, right? And an important concept here is therapeutic optimization and individualization, okay, which is really all about dose. This isn't new. Even our former director coined the mantra, the right dose, the right drug for the right patient at the right time. And this is all about finding the right dose across therapeutic areas in drug development. And so a legitimate question that people have is, so why are we so excited about this now in oncology? Well, what's changed to make us get very excited about this and launch this new initiative? Well, this is a slide that I borrowed from Dr. Lanre Okusani in our group, and I think it underscores the reason for this. So what we have here are the drugs that were approved over a 7-year period in oncology. And all of these drugs had PMRs or PMCs for these products to find the right dose, either for a special population or the general population, after the drug was approved. Okay? And the examples that are highlighted in red are examples of drugs where we found the dose was too toxic in the patient population, and we needed to find a better dose with less toxicity after approval. And that is a very difficult thing to do. We've been successful in some cases, and others not so much. And if we take Mylotarg as an example, that was a drug we originally approved in, I think it was 2000, but it was too toxic and it came off the market. And it wasn't until 2017 that that drug was reapproved at a different dose and a different regimen that alleviated a lot of that toxicity. So it's not easy to do this in the post-approval setting. And so the whole point of what we're trying to get at here is try to push that effort earlier in drug development to make better decisions, or at least the best decisions we can make early in development about the optimized dose of these drugs. So I want to go through a case study here. I want to look at idelalisib to look at some of the things we might have learned during development. And before I get into this, I want to point out that the purpose of this isn't to excoriate the folks here or say they did a bad job. Absolutely not the case. They actually did a lot of good things. They've done some things that their peers haven't done, and I'll point those out along the way. But this is a very good case in my mind because it's a recent example, and it has a lot of characteristics of other drug development programs that we have seen. Okay, so I think it's worthwhile to go through this and see what are the things we might learn from this, looking back with 2020 vision. So this drug was approved by the accelerated approval pathway in 2014, right, for 3 different indications. And this indication shown here is CLL. Now, they did a randomized controlled study, which is a very good thing, using progression-free survival as the endpoint. And what we saw was the PFS was much better in the idelalisib arm, and you can see that the hazard ratio really favored the use of this drug. And so that was very promising. Okay. And then for the other 2 indications, follicular lymphoma and SLL, these were single-arm trials that were based on ORR. And here again, you can see that the response rates were 54 and 58%. Again, that looks very good. So very, very promising. But then as a part of the regulatory approval, they had to do their confirmatory trials. And in 2016, we got kind of a shocking result, I would say. As you can see in these 3 trials, they had to be terminated due to an increased number of deaths in the idelalisib arm. So if you look across these trials, you can see the rates of death were higher than the control arms, and the hazard ratios were much higher, indicating that we may be causing harm by using this drug. So this is sort of a shocking result. Now, when we look at this a little bit further, you can see that the adverse events for idelalisib were much greater than the comparator arm for things like grade 3 infection, diarrhea, colitis, AST, ALT, and pneumonitis. So they appear to be getting more infections using this drug. Now, another way to look at this is to look at what happens to the dose modifications. So this slide is a little complicated, but it shows us dose interruptions, dose reductions, discontinuations, as well as serious AE and grade 3 or greater AEs across these trials. And idelalisib is shown in blue, and the comparator arm is green. And just looking at this quickly, you can see that the events occurred at much greater rates with idelalisib, and in some cases, they were much greater than the comparator arms. So again, it's underscoring that there's problems with this. There's some toxicity associated with this drug that is very problematic. Well, at the end of this, we're sort of sitting here scratching your head, asking ourselves, where did we go wrong with this, right? We'd seem to do everything well, and we had good early indications. Well, let's go back and look at the beginning. What did they do at the beginning of drug development? How did they select this dose? So this is a schematic that shows you the dose escalation and expansion trial they did, okay? And they did a lot of good things here. So first off, they looked at 3 different regimens, okay? They looked at BID, they looked at daily dosing, and they looked at a BID every 21-day cycle. So that's a good thing. We often don't see in other programs. It wasn't long before they discarded this particular regimen and the daily dosing regimen and went with the BID regimen, okay? Now, another thing you can see is each of the dose levels, they've actually enrolled quite a significant number of patients. We're often in the case where we see drug development, and because we're moving hard and fast, we might only see a couple of patients in lower dose cohorts, okay? And maybe that's not so informative about these lower doses. Now, the purpose of the trial was to find the MTD, but they didn't achieve that. What they found, though, was when they got to about 200 or so, they started seeing a lot of grade 3 events and higher, okay? So that compelled them to select the 150 milligram BID dose, and they then did a dose expansion in patients to get a better characterization of that. So they've done some very good things here. One of the other points I should point out, this is done in patients with a variety of hematological malignancies. But what did we see in terms of that data early on? Okay. So if we look at the efficacy, one of the interesting things here is we have the outcomes in some of the relevant patient indications across the doses, okay? And if you looked at 350 milligrams, you're looking at like 6,700, 70% response rates. And you're, wow, that's pretty good. You know, even at 100, it's also fairly high, but they didn't select these doses. That's really going to be about toxicity. I'll come back to that in a moment. So they ended up focusing on 150 because they were getting good response rates there. Okay, makes sense. But here's the interesting thing, I think. If you look at 100, they're also getting some pretty good response rates. So it makes you wonder why we weren't looking at 100 a little more closely early on, because they seem to have pretty good response rates. Well, if we go to the safety part of this, you can see that these are grade 3 events. And if you look at the 350 and the 200, you can see we're looking at 18, 20% occurrence of these events, and that's too high. So that's why they didn't pick these higher doses, right? They went with the 150, and you're hovering somewhere around 10, 11%. And on the surface, that seems pretty reasonable. But again, look at the 100 milligram dose, okay? You have a reasonable number of patients. And you're looking at around 8%. So the occurrence of grade 3 events seems to be lower. So again, it begs the question, why didn't we think about the 100 a little more? Okay. Well, moving along, when we got the NDA, this is some of the information that we had, okay? And I think it's a little instructive. So this figure here shows us the C trough concentrations for the different doses of the drug. And you can see they're above the IC90 for the PI3K delta enzyme, okay? So at the 150 milligrams, you're above that. You're also above that at 100. And arguably, maybe even 50, you're above that IC90 most of the time. Okay. When we look at the biomarker, the lymph node reduction, what you see is it's pretty much a flat response, okay? Now, maybe that's not surprising because I think this is mostly with the 150 milligram dose, so you don't have a super wide range of concentrations, but there's really no hint of an exposure response here. Okay, so moving on, if we look at the efficacy outcomes, you can see for progression-free survival that the exposure quartiles keep crossing over each other. So indicating we're not really seeing any exposure response. That's the same in the CLL trial. And it's kind of an indication to us that we're probably on the flat part of the dose response curve for efficacy. Now, if we go on and we look at the exposure response for safety, at least for some of them, we're seeing a relationship between exposure and outcomes, right? So for grade 3 diarrhea, as exposure goes up, dose goes up, we're going to see more events. Conversely, if we reduce dose and exposure, we're going to reduce seeing some of those events. So where does that leave us? Okay. So what we did, we went after the MTD and it wasn't reached. I think this is one of the important points. Okay, I think by going to the MTD, we weren't thinking as much about lower doses. We know that the exposure response for efficacy was flat, right? But we still are seeing some rising toxicity in relation to dose and exposure. And some of the non-clinical data and some biomarker data is underscoring that, 100 might actually have been a viable dose to look at, okay? So why didn't we? Well, I think that's summed up in this figure, okay? So this curve on the left here shows us the sorts of exposure response relationships we see for toxicity and efficacy with cytotoxic drugs, right? They often parallel each other. And it's often very difficult with these kinds of drugs to distinguish between the efficacious and the toxic dose, okay? And so based on this, people have pursued the maximum tolerated dose. Okay, we're trying to maximize the amount of efficacy we can get because this is a life-threatening disease. The downside of that, the problem with that, is that we're going to sacrifice the toxicity part of the equation here. Okay, so you often have doses that work, but you have high rates of toxicities as well. However, I think most of our drugs now, we're using a lot more targeted drugs, are in a different situation. Okay, and I think this is a curve or a set of curves that reflect the reality we see here. There's some other variations of this, of course, but generally speaking, we probably more see this sort of exposure response for efficacy where we're coming up and we're plateauing at a dose, whereas some of the toxicity curves are still on the rising portion of their curves as the doses get higher. Okay? And I think this is the case for idelalisib. I think we're somewhere in here. We know that the efficacy is plateaued. But what this means is for these drugs, we can probably afford to reduce the dose here a little bit without significantly impacting efficacy too much. But that could have a big effect on the toxicity, all right? So that's why we're saying it's more important. We need to think more about optimizing the dose rather than just going after the MTD. So if there's one thing you remember from my presentation today, I'd really like everybody to start thinking more about dose optimization and less so about MTD. There will be some cases where it's still the right thing to do, but we have to think about this more because I think when we're pursuing the MTD option, we're thinking about getting as much drug in there as possible. And we're not thinking about the other possibilities where we can still have an efficacious drug at a lower dose with less toxicity. Okay? And I think that might be some of the things we saw going on with idelalisib. Another point for you to remember is we generate a lot of information in the development of our drugs. You know, what is that? Oops, real quick, sorry. What does that information tell you, right? Let's maximize the use of that. And so we've talked about some dose optimization strategies before. This is not an exhaustive list, but these are some of the things you can think about. MTD, let's try to get away from that as the goal for dose selection. Think more about, or make as much use as you can out of your non-clinical data. What's it telling you? How does it guide you in selecting your dose? Enrolling sufficient patients to characterize the kinetics, the efficacy, and the toxicity. Idelalisib did a lot of good work here. Okay, they got a lot of information, more so than other cases that we've seen. Okay, but this is very important. And another important point here to remember is you need to enroll a sufficient number of patients in the indicated population to get a good handle of the behavior of the drug to help select those doses appropriately for those populations. Excuse me. PK-PD relationships, also very important. And in a moment, Hao is going to wow us with some examples of MIDD. When we get to the point where we've done our dose escalation and we're going to expand the dose cohort, it's probably a good idea to expand a couple of dose cohorts so we can get a firm handle on the behavior of the doses we're looking at. Oftentimes, randomized parallel dose response trials might be the best way to determine the best dose for your drug. And other examples or other possibilities could be to use multiple doses in the registration trial to get all that kinetic and efficacy and safety data. So I'm going to stop here. I'm going to hand this off over to Hao now. I'm going to go back to Arlene because she has another poll question for you, and then we'll segue into Hao's presentation.
Thank you, Brian. And yes, you've pitched a great question that we should ask the audience. So in dose escalation trials, how often do you use optimal dose as the goal? And we'll give you a few seconds here to go ahead and answer. Again, I'd like to remind you all that we have over 1,000 live attendees today. And while we thank you for spending your time with us, I know you all have questions. Thank you for popping them into the Q&A panel. So we won't be taking questions or unmuting or on the phone today, but go ahead and put them into the Q&A tab and we will get those questions addressed or followed up with. So I'm going to go ahead and end the poll here and we'll share those results. So it looks like more than half the time is leading. And with that, I'll have Hao lead into his presentation. Take it away.
Sure, thanks, Ellen, and thanks, Brian. So I'll be happy to continue the conversation. I'm sorry, can we go back? I'll be happy to continue the conversation on how model-informed drug development, or MIDD, can be applied to support drug development. Especially, I would like to encourage the community to work together to facilitate dose optimization for oncology drug product development by using MIDD tools. So next one, please. As we know that MIDD begins as a concept, as a technical concept, it really refers to the development and application of exposure-based biological and statistical models derived from preclinical and clinical data sources to address drug development or regulatory issues. And over the past decades of practice, the community has developed multiple tools. So MIDD begins with PK models. For example, we know POP PK models and PBPK models. Then it gets extended to the exposure response models. And further, some disease models were developed to characterize disease progression and patient characteristics. And this information can be applied to conduct in silico clinical trial simulations and improve clinical trial design. And in recent years, novel techniques, including quantitative structure and activity relationship, or QSAR models, systems pharmacology models, artificial intelligence, or machine learning models are used to further expand our capacity in using modeling and simulation tools to streamline new drug development. As expected, the most common use of MIDD approaches is dose selection and optimization. In addition, MIDD approaches have been used to allow pediatric extrapolation, improve clinical trial design, identify new clinical endpoints, and enhance patient selection and enrichment. Over time, we may see additional applications of MIDD in new drug development. So we are sure that this technique will bring more capacity in oncology product development, and especially for dose selection. Next one, please. To allow dose selection and optimization, we should consider multiple factors. For instance, we should consider the mechanism of action, the physical chemical properties of the compound, the formulation characteristics, the learning from preclinical data, existing knowledge of the disease and patients, the prior understandings and clinical experiences of compounds with similar mechanism of action, and the features of PK/PD for the compound under development. And therefore, dose selection can be a complicated process that is built upon integration of knowledge with various sources. So if we really consider that MIDD is a machinery, really provides a unique platform to integrate all pieces of information and existing data together. As a quantitative tool, different MIDD approaches, as some of them are listed here, provide different angles to combine data together. The MIDD tools may generate information through in silico trial simulation or obtain information which may serve as evidence for dose optimization and selection. So next one, yeah. So besides these technical tools, we would like to remind you that there are regulatory tools that you may consider when you have questions regarding dose selection in oncology drug product development. And several avenues are open to promote earlier interactions between sponsors and the FDA on the use of novel approaches for new drug development. And these programs have been and will continue to be used to support Project Optimus for oncology dose selection. For instance, the Fit for Purpose, or FFP program, is intended for tool development. Novel modeling tools that can be generalized to support different new drug programs should be discussed under this program. whereas MIDD paired meeting program and CID program are designed more for specific drug development. The CID program focuses on efficacy determination. For instance, the general topics may be related to the application of a Bayesian approach to streamline a drug development program. MIDD paired meeting program is designed to address various topics that may encounter and different phases of new drug development. In the next few slides, we're going to talk to you about some of the details for the FFP program and MIDD program. Next one, please. Well, let's start with the Fit for Purpose program. This program is intended to provide a pathway for regulatory acceptance of modeling tools that may be useful for different drug development programs, sharing some common features. An applicant may submit the modeling tools, the supporting documents, and the data. A designation of fit for purpose will be established at the outcome of the review process. So far, we have granted 4 modeling tools, and some of which are summarized in the table to the right-hand side of the slide. The first submission was a disease modeling tool for Alzheimer's disease, and the rest of the 3 other tools that can be used to support those findings that we have listed here in the table. For instance, in recent years, Dr. Yuan from the University of Texas and MD Anderson developed a Bayesian-based tool that can be used to determine maximum tolerated dose, or MTD. And this tool is called Boyne and was granted fit for purpose in 2021. Likewise, Pfizer developed a Bayesian Emax model, which may be used to facilitate a dose selection. And this tool was determined fit for purpose earlier this year. And all these tools can be useful for oncology new drug development, and hence be applied to support Project Optimus. And we hope to review more innovative modeling tools from the community to modernize dose selection and other aspects in new drug development for oncology and other disease areas. And next one, please. MIDD paired meeting program is another important program. This program is jointly administered by CDER and CBER. The Office of Clinical Pharmacology is a point of contact. To qualify, the applicant must be a drug or biologics developer with a product that is registered under US IND, NDA, or BLA. The applicant may submit a meeting request over the year, and the FDA may grant a defined number of submissions on a quarterly basis. You can see the 4 little guys holding different pieces of puzzles and putting them together, as shown in the figure to the right, really represents the nature of the meeting. The meeting represents a joint effort from all stakeholders in new drug development and from multiple disciplinary review team members. The pilot program was initiated back in 2018. And based on accumulated experience, we expect to continue this program in PDUFA 7 as a formal review program. Next one, please. Well, this slide summarizes the current experience from the MIDD parent meeting pilot program. As shown in the figure to the left, there is a clear increased number of meeting requests over the past 4 to 5 years. Of course, the summary for year 2022 is incomplete at this point. So back in 2018, the total number of meeting requests was 7, and we granted 6 of them. In 2021, the meeting requests increased to 18. We granted 9 for face-to-face meetings and 3 for written responses. Click once, please. The figure to the right shows which disease areas that MIDD paired meetings were held. The largest number of submissions is from oncology and followed by neurology and infectious diseases. And most of the submissions focus on dose selection and dose optimization. With a general understanding of MIDD as a technical concept and relevant regulatory tool, I'm going to switch gear and talk about some case examples. Next one, please. So the first case is called osimertinib. Next one, please. Now, osimertinib is a 3rd generation of EGFR kinase inhibitor for the treatment of non-small cell lung cancer bearing certain mutations. The approved dose is 80 milligrams given orally once daily with or without food. This program has undergone extensive preclinical and clinical development. So following initial accelerated approval, the FDA granted a regular approval. Next one, please. And preclinical stage, the compound was developed based on a thorough understanding of the underlying disease and the mechanism of action. The mutation that is account for drug resistance to the earlier EGFR kinase inhibitors became the target for the design of osimertinib. The preclinical evaluation using the specific cell line models, xenographic disease models, transgenic mouse models, and patient-derived explant models provide solid evidence on the effect of the drug. The quantitative findings provide a general sense on the possible target concentration range. Next one, please. Preclinical data demonstrated the suppression of tumor growth and different dose and concentration levels. For example, as you can see the figures to the right-hand side of the slide, demonstrated the tumor volume change over time following different doses of osimertinib with the different tumor models. Clearly, there is a dose and exposure response relationship. Doses greater than 1 milligram per kilo for PCA9 model, which is exon 9 deletion, and show tumor volume reduction over time. Likewise, doses greater than 2.5 milligram per kilo show tumor volume reduction in H1975 model with L858R and T790M mutation. Next one, please. Our PK model is established to describe the concentrations of the parent compound and the metabolite. Subsequently, the concentration data are linked to the PD marker change and tumor volume reduction. This model allows a test of tumor suppression effect for different tumor mutants. The modeling results support further clinical development of the drug and predict that the first human dose can be started from 20 milligram. Next one, please. So the phase 1 clinical trial, AURA, was thus conducted over a broad dose range covering from 20 milligram to 240 milligram. So even though the patients were not enrolled through a mutation status, the existing data allow a direct comparison of the treatment effect in patients with T790M mutation. Clearly, there is a good response in this patient population over a wide tested dose range. It is also noted that doses above 80 milligram are less tolerable due to increased incidence in skin disorders, nail effects, diarrhea, severe grade 3 AEs, and more frequent dose reduction. Next one, please. So the drug was further tested in the expanding phase of AURA and AURA2 trials. So over the tested exposure range, the exposure response relationship for efficacy appears to be relatively flat, whereas the ER relationship for safety for rash and diarrhea appear to be relatively steep. Next one, please. So all these findings are confirmed by AURA3, a phase 3 clinical trial to compare the treatment effect between osimertinib given at 80 milligram once daily as compared to chemotherapy. And the primary endpoint is progression-free survival. The ORR and duration of response were also assessed. And there is a 6-month difference in median PFS between the treatment arm and the comparator arm. The ORR has been doubled in treatment arm, and plus the median duration of the response is 6 months longer in the treatment arm than chemotherapy. Next one, please. So through the case study, it shows that dose selection is an integrated process, and modeling simulation tools are helpful to combine different pieces of information together, including dose, PK, PD, and clinical responses, so that the findings from earlier phase development can be translated to inform dose selection and clinical trial design in late phase. And the robust dose or exposure response relationship on both efficacy and safety may provide a solid basis for dose selection in general patient population and patient subgroups. Next one, please. Now we're moving to the second case for drug X. Next one, please. Drug X is a bispecific antibody under clinical development for the treatment of hematological malignancy. And this diagram shows the structure of the antibody. And one FAB arm targets CD3 T cells, and another arm targets a specific tumor marker. A common issue for bispecific antibody treatment is the potential for developing cytokine release syndrome, which depends on the severity and sometimes can be life-threatening. So to minimize the risk of developing severe cytokine release syndrome is the key issue for developing the dosing regimen for a planned phase 3 trial. And therefore, modeling simulation was applied to evaluate alternative priming dosing regimen with the objective to minimize the risk for developing severe cytokine release syndrome in the meantime to achieve sufficient anti-tumor effect. Next one, please. So a complicated modeling approach was developed by the sponsor. A population PK model was developed to generate the PK profiles with the variability under different dosing regimens being tested in the clinical program. A QSP model was used to link the concentration change of drug X and relevant changes in IL-6. A subsequent exposure response relationship was established to describe the potential of developing cytokine release syndrome with different grade levels based on the various levels of IL-6. The cumulative events of cytokine release syndrome can be calculated. In the meantime, a separate exposure response model was developed to link drug X concentration with the temporal changes on tumor size. And next one, please. So based on the established model framework, the effect of various combinations of dose and dosing frequency as the priming dose can be assessed through clinical trial simulation. The outcomes for the associated changes in suppression of tumor growth and incidence of and severity of cytokine release syndrome can be compared in order to drive the optimal dosing for follow-up clinical trials. So next one, please. So with that, I'm going to conclude my part of presentation. And I have the following take-home messages. First, dose optimization is critical in new drug development for oncology products. And second, Project Optimus in combination with other programs such as MIDD fit for purpose provide an essential pathway for earlier interactions with drug developers to improve dose selection. Third, quantitative clinical pharmacology tools can be broadly used to support dose optimization in new cancer therapy under development. And next one, please. And Brian and I have a long list of individuals who have contributed significantly to the review cases and the presentation. Last but not least, we would like to thank you, our audience, for your attention. We'll probably bring one more poll question, and after that, we're open to questions and comments. So Arlene, are you going to pull out the poll question?
Yes, thank you, Hao. Excellent presentation.
Thanks.
So our final poll question today is, how often do you use MIDD for dose selection? And we'll give you a few seconds here to go ahead and answer that. While you're putting your answers in, Tom Sun will be getting ready to get the Q&A session started. If we don't get a chance to address your specific question, we'll move it over to LinkedIn, where everyone can continue to post, and we will tag both Brian and Hao and Tom in the chats, so that way you'll be able to continue the conversation on this very important topic. I'm going to end the poll now and share the results. So it looks like more than half of the time. And with that, Tom, I'll go ahead and hand it over to you. Take it away.
Thank you, Arlene. And first of all, I'd like to thank Brian and Hao for outstanding presentations that really provide a lot of insights for this important topic for Project Optimus. So far, we have 42 questions posted in the Q&A tab. I'm trying to do my best to kind of look through the questions and maybe post some common ones and or interesting, important ones for consideration for our speakers. I think one of the questions very practical and that people will be asking it on the regular basis is that once the monotherapy trials, the dose optimization is completed, and the sponsor move on to the combination therapies, what is your view and general suggestions for the sponsors? How do we learn from the monotherapy trials, and do we need to optimize each of the combination therapy for dose optimization? Thank you.
Yeah, that's a good question, and it's one we're doing a lot of thinking about now, and we're delving into some of the ways to go about addressing that. I think some of the things that we have to think about, or I think the likely scenarios that we'll be faced with are we're going to have situations, I think, where it may make sense to take the approved monotherapy dose into a combination and move ahead as is. But there are probably other cases where we're really going to need to do some dose exploration with the new drug because of toxicities and so forth. And in fact, you might end up in some situations where we're going to have to vary the doses or do some dose ranging with both the new therapy and the combination. And all of this is really going to depend on sort of the disease setting you're looking at and the stage of the disease and those kinds of things, the setting that you find yourself in, overlapping toxicities. It's a complicated situation, and I don't think there's a one size fits all. We're going to have to look at, I think, each case based on the intrinsic behavior of the drug and the disease setting and those kinds of things.
Great.
Just to add to what Brian just alluded earlier, I think this is an excellent question. We've been talking to quite a few scientists internally meaning within FDA, and externally with the sponsor and with some of academia people, and try to look for the possible pathways to derive optimal doses for under combination scenario. I think there's several interesting ongoing research to our knowledge that is ongoing, and maybe some of the modeling, especially mechanistic modeling, can potentially provide some insights. Maybe that's the area that I would say the community can actually work together as a joint effort to see what would be the best approach to derive the doses for combination therapy, given the understanding even for monotherapy, how we can actually find it in an efficient manner, and also find the dose that is optimal in terms of minimize toxicity and also bring the most benefit of efficacy.
Great, thank you both. And maybe a follow-up question related or not related. There are a couple of questions related to application of PBPK type of modeling into the general approach for MIDD for Project Optimus. And the reason is that a lot of times for oncology patients, the ability to get PK samples will be really difficult, and particularly during the COVID time that sponsors may experience difficulties for patient access clinical sites and things like that. And so could the PBPK approach be applied in the general approach if somehow the exposure data are difficult to get? Thank you.
I think that maybe I can start. Brian may just chime in. So my short answer is yes. Yeah, PBPK approach can always be used to drive some critical decisions, especially for dose selection, and we understand the challenges that actually exist in oncology setting, and also especially under the COVID. We're having challenges to enroll patients in the trial. So PBPK modeling may provide a lot of insights. Having said that, all this PBPK results should be actually evaluated on a case-by-case basis, because there are a lot of nuances in terms of how do we look at the PBPK modeling, right? So essentially, we can look at the assumptions, we have to look at the source of information for you to borrow information from, because a lot of times PBPK modeling we have to use other sources or alternative sources to get the data from, or to get the parameter estimates from. So I also have to look at what is the utility of PBPK modeling. So is that used for trial design, or this is really used to replace clinical trial? So there are different standards applied to accept the PBPK results. Having said that, I think even under oncology, we have used the PBPK modeling for some of the critical decision making. I don't know if Brian, you wanted to allude it further on that.
No, I don't think I can really add anything more to that response, Hao.
Great, thank you. And I think there's a list of questions all related to the first example, Brian, you presented at the lectureship. And the questions are related to some of the dose escalation, dose ranging information are coming from very narrow dose escalation range, right? 100 BID, 150 BID, and the PK data may appear to be very overly overlapped. And so to make certain conclusion based on this kind of data, could you comment maybe from Brian and for how to comment on that? And secondly is that there are a couple of questions related to the sample size that I think the agency recently in March 2022 has some guidance about the early clinical trials in terms of expansion phase, sample size considerations, and may or may not to kind of suggest a cap for certain cohorts numbers. And could you also give some perspective on that as well? Thank you.
Okay, so for each drug, when you're doing a dose escalation, you want to try and look at a reasonably wide range of doses to get a good understanding of, as I mentioned, the PK, the efficacy, and the safety events. And I'm not sure that there's really a hard and fast number that we can use about that. I think oftentimes we do see some overlap in the kinetic exposure. But that's, I think, where it's important to get additional information around the efficacy and the safety part of it to get a better completion or a more complete picture of the behavior of the drug. I don't know if Hao wants to add any perspective to that part of the question.
I think Brian, you have covered the main thing. So I think is about the same. I think we needed to have explore a broad range of doses and or exposure ranges that really provides us a lot of information about what we can further either put in the model or use that information to guide a further trial design. And if you studied a very narrow dose range or a very limited number of patients in the very small dose ranges, that doesn't really give us a lot of information on either toxicity or efficacy of the drug. That's not going to be easy for us for you to determine for dose.
Great, thank you. Next question, maybe the music to your ear for both of you. Would FDA make another options for agency sponsor interactions with a sponsor to help the companies to do the dose optimization? And what's your recommendation of which stage of drug development that the sponsor should approach you, or for agencies to kind of sponsor such interactions with the sponsors?
So whether or not there's going to be a specific time point or process for addressing this is a matter we're dealing with internally, and there will be more on that in the future, I think. But I would say in reality, we're already starting to do this. We have been doing this. So when people are coming in early in their drug development, their phase 1 trials, and the phase 1 trials, before they're going into phase 2, we're already getting into discussions with various folks from various different places about the dose selection processes that they're going through. We're asking questions and getting into discussions about what the data means and what other things we may need to look at. So those kinds of things are already going on. So that's that part. Was there another? I think there's another part and I'm forgetting it. Was there another part to the question, Tom?
Do you have a plan to have agency sponsor events or mechanism?
You mean
generally is sponsored to approach you hey, we want to discuss phase 1 conversation. Do you have any?
Well, in terms of the drug development part, when people come in with their applications, we have those discussions. Other things you'll probably see in the future will be more public types of interactions in various different fora to interact with folks in various different disciplines and cross-discipline areas to discuss these various issues. I think you'll see a lot more of that in the coming year or so.
Great.
Yeah, I would like to add what Brian just alluded to. So I think a lot of times, I think the sponsor really knows their product better than any of us. So then it's the perfect timing to reach out to us if they have issues or questions about the dose finding. And my general sense is that it would be a good idea to interact with us earlier rather than, because you can imagine that if you're really flying an airplane, you're just 5 inches above the runway, you're about to land. There's not too much maneuver we can do here, right? Either from your side or our side. So it would be better off when you just start the journey actually on the flight and actually in the air. So there's probably more things we can do together.
Great, thank you. And next question, slightly related to this topic. Also from your presentation, Brian, that you use the example IC90 value for PI3K inhibitor in vitro assessment. And could you elaborate that the agency's view that how generally that you see using this kind of preclinical in vitro assessment to position the clinical dose, and how do we harmonize the information from the clinical data in terms of fresh PK/PD data to I think that's a very, very common situations that all the sponsors are asking.
Yeah.
Yep.
So my perspective on this is we can use the preclinical data, non-clinical data, whatever you have, to guide us. It sort of gets us into the right ballpark, right? So if the IC90 was the important parameter related to the activity of the drug, if you were below those concentrations at a particular dose, then that is probably a good indication that you're really not close to being where you need to be. Whereas if you're maximizing the effects, then that's probably telling us we're in the right ballpark. We need to be looking around these kinds of doses. Okay, I don't think that we can use that information in and of itself to make a dose selection. I think it's very helpful to help guide us. Okay, and I think it sometimes provides us a little extra insight in conjunction with the patient data that we get on the kinetics, biomarker data, and outcomes, efficacy and safety and things like that. So I think it's an important part of the puzzle. I think we should make use of that as much as we can. Hao, did you want to add anything?
No, I think you have covered.
Thank you. So following on the earlier question that also related to your March 2022 guidance in terms of a sample size, that it specified the recommendation that the cohort sample size is about 20 to 40 per indication. And so are you suggesting that to have multiple dose kind of dosing ranging evaluation per indication, or what is your view on kind of seeing this draft guidance and giving us some suggestions, thinking about study design perspective, some general thoughts? Thank you.
Yeah, so generally speaking, these are my thoughts. Other people have other perspectives on this that are valuable as well. But, I think often what we have seen in the past with dose escalation is, we're pushing very hard to get to the MTD. Time is money, we need to try and get this figured out as quickly as possible. And certainly at the lower doses, we have seen dose cohorts with only a couple of patients in there. Okay. And that may make a lot of sense early in the development phase. We often don't anticipate seeing a lot of activity at the lower doses. But then as we push higher and we get closer and we get more information about the doses that we're interested in, then I think we've got to start thinking more about expanding those dose cohorts so we can get better information around the kinetics, the efficacy, the safety, so we can make better decisions early on about what that right dose looks like. Okay, we have a lot of examples in the past where we've had a very truncated dose escalation scheme. There's only been a few patients in the dose cohort, and people thought they got to where they wanted to be, and they put all the effort in that one dose cohort, right? And so you have information for that one dose course, and not so much around some of the other dose cohorts around it. Okay, and I think that's often put us in positions where we found out later the dose wasn't quite right, and we probably would have liked to have that information earlier in development. And so that's my perspective on that particular part of it. How did you want to add to that?
Yeah, I totally agree. I think just looking at a broad range and also expanding the cohorts, or not just one, maybe just multiple cohorts, would be very helpful to really define the efficacy and safety profile of the product, of a compound. That would be very useful to determine the final dose in general.
Mm-hmm. Thank you. And we are 1 minute after, but there's one very interesting question from audience that in terms of application of Project Optimus, in your view, does that adding time to drug development process or saving times? And Brian, you've been seeing several hundred applications and helping a lot of sponsors. What's your general view on applying this kind of approach to help sponsors?
Yeah, clearly I think it's going to. We're going to spend more time in the early part of development. Okay, we have, when you're pursuing the MTD, we're pushing through doses very quickly until we can get that highest point. If we start to look more and investigate doses to make better selections about optimization, it's going to slow us down a little bit at the front end. And there's no escaping that. That's the reality. But I think it's the thing to do, because we had found in many cases after we improved the drug, it was too high, right? And then it becomes very difficult to correct at that point. So I think that the effort we put in at the front end is going to benefit us in the long run. And we really think that this is where we should be spending a lot of effort.
Great. Hao, want to add anything?
Yeah, I totally agree. I think it's probably a time or a cost that probably add a little bit to the development program, but it will turn out to be all worth it, because if you end up landed to a wrong dose or a dose that's not quite optimal, it's going to cost the sponsor also, because you're making your product less competitive to another product which has maybe similar mechanism of action. But just a better profile, safety profile, bring their market, a priority, market advantage too, because they can gain more market share possibly. So that is also to the benefit of the sponsor, and of course, definitely to the benefit of patients. We needed to find optimal dosing too.
Great, thank you. One final question for short answer. Can the sponsors still submit MIDD program at the words is the pilot program may end? Are you gonna make this a more permanent base?
Yes, yes, definitely. Maybe I can start here. The short answer is yes. We are planning to extend this program as a formal review program. So we plan to probably send out some official notice during the first quarter of the fiscal year of 2023. So just you probably want to look and watch out in the notice that we're going to send out. That's going to lay out what is going to be different or maybe similar across the pilot program and the formal program.
Great, thank you very much. We are 4 minutes over the top of the hour. And so I'm gonna pass this back to Arlene to have a closing. But before that, really like to thank Brian and Hao for your outstanding presentations and very open dialogues with Q&As with the audience. Thank you.
Thank you, Tom. It was our pleasure.
Yes,
Slide,
again.
Ken.
Thank you.
Yeah, thank you, Brian and Tom. We appreciate you sharing how Project Optimus combined with MIDD can provide an essential pathway to improve dose selection. And as Brian said, time is money, so we will let everyone get back to making that. On behalf of everyone at Simulations Plus, we invite you to learn more about our MIDD solutions at www.simulations-plus.com. This is also where you can find a copy of today's slide deck and webinar replay. This concludes our webinar for today. And Brian and Hao, I hope we see you back here again soon. Thank you, everyone.
Thank you.
Thank you.
Thank you very much.
Bye.
Bye.